La diffusion vient de démarrer. Tous les participants sont en mode écoute seul.
Hi everyone. Thank you for joining us for this third webinar. It's going to be very exciting. I'm Elena Legrand. I'm one of the committee members of the EMN Early Career from the Metabolomics Society. So I think we can start. It's time. Everybody's here, or almost. So just before talking about the webinar, if you want to connect with us, just as a reminder, don't forget about our Facebook page, or Metabolomics Twitter, Metabolomics Society and LinkedIn, also on the forum. And then you can join us by email at info.emn@metabolomicsociety.org. So also, just for the one of you that would be late, think about it. The poster abstract are due on the 26th, so it's coming soon. So don't forget about it. And finally, today we are featuring Dr. Hiroshi Tsugawa from the Riken Institute in Japan. So we are very excited to have him with us today. He's going to talk about computational mass spectrometry in metabolomics to deepen, the understanding of metabolism. Yeah. I think something went wrong. Hopefully not. So I'm gonna give.
You're good.
Sorry.
You're good, Elena. Keep going.
Okay, cool. So something went wrong on my screen, so that's okay. So I'm gonna let Hiroshi tell us everything about his talk.
All right. All right. Probably, I hope you can see this screen. And so good morning, and actually good afternoon and good evening in many countries. I'm Hiroshi Tsugawa of Riken in Japan, and Japan is currently 8 a.m. And thank you so much for Europe audience in this very late time. And thanks for US area audience in this busy time. And today I would like to talk about the importance of computational mass spectrometry in this talk. But first of all, I'd like to say thank you so much for Elena Legrand and Early Career Members Network for Metabolomics Society to give me this opportunity. So, and also I'd like to appreciate the main collaborators for my study here. And the above photo is the main members for plant metabolomics that I will present today. And especially Ryo Nakabayashi is the main collaborator of Kazuki Saito's group. And I also appreciate my direct boss, Masanori Arita, for giving me active science opportunity. And the middle photo is a great laboratory that I've ever belonged to. And I studied many things for metabolomics here and appreciate my US teachers, Oliver Finn and Tobias Kint and Tom Chaika. And the last photo is the laboratory that I recently started to join to learn the biology of lipids. And I appreciate Makoto, Arita, and the lab members to perform the active discussion for medical sciences. And actually, I'm this guy. And this is just a brief self-introduction. And please contact me in some conferences if you have an interest for my study. Okay, let's move to the science talk. And I'm using this kind of slide to explain the reason why I'm doing computational mass spec. So as you know, we are supposed to analyze very crude biological samples in Metabolomics, and we will find many peaks like this. And we are supposed to make this kind of data matrix which contain the metabolite name and sample names, and so a kind of ion abundance information in each cell to perform the statistical analysis like PCA or PLS. However, so at least when I was a student, so it was very, very time-consuming tasks to create this kind of data matrix because few software programs are available for metabolomics workflow in those days. So it means, so even if the biologists have a nice idea and even if they bought very high quality mass spec, so many researchers would disappoint to the complexity of metabolomics data. And therefore, because I was in the bioengineering department and I saw that it's important science to create the automated system to deal with the metabolomics data from mass spectrometers. Also, therefore, so far, I developed the several software programs which are currently available at Riken Prime website. And so of this, so probably MS Dyer and MRM probes are the most universal programs which support all of metabolomics workflow from raw data import to statistical analysis. And of this, so MS Dyer was optimized for untargeted analysis, and MRM probes was optimized for targeted analysis. And these programs can import mainly 3 data formats, CDF, AVF, and MZML, although the AVF format is only available at my software program. And in untargeted analysis, so MS Finder was also developed to elucidate unknowns, so for unknown spectra in GCMS and LCMS projects to predict molecular formula and metabolite class and structures for unknown spectra. And so once the first screening is finished by using the MS Dyer program, so users can use the MRM probes for the targeted approach because this graphical user interface was optimized for metabolite quantifications. And finally, there are several programs to perform, for example, data normalization and multivariate analysis. Also, so recently, so these programs are already integrated in MS Dyer program. And there are also several specialized tools, so which were developed for the specific study cases. But I think MS Dyer is the biggest program, and there are many users for use this program. And therefore, I just would like to introduce the main collaborators for MS Dyer development. And there are 2 main developers who are writing the source code. And Ikuta Tada is a student of National Institute of Genetics in Japan, organized by Masanori Arita. And his main work is to develop the function to deal with data-independent MS-MS acquisition with the team of Dr. Greg Wheelock of Karolinska Institute. And he's also developing many useful graphical user interface for GCMS and conventional LCMS project. And Diego Pedrosa created the function to link the MS Dyer with Mass Bank of North America. And Tom Chaikar, he was originally in Oliver Finn Lab, and he is now working at Czech Republic as associate professor, and Romanas Haleskis, he's an active collaborator of IPUTAR at Gunma University, and they are a kind of the technical advisor for us to make our program more effective. And of course, so many other researchers and RECAN staffs are currently contribute to the MS-style program development, and I would like to appreciate all their contribution to improve MS-style program. And today, I'm actually talking about our latest achievement tackling the comprehensive annotation of plant-specialized metabolite. But in this slide, I'd like to state that the science of computational mass spectrometry can be applied to various fields like lipidomics and primary metabolite profiling. And our technique can also be applied to several new technologies of mass spectrometry, which includes ion mobility path acquisition method and new fragmentation methodology, something like this. And I will give the presentation about it in Metabolomics conference of this year. Also, our technique can be applied not only for metabolomics, but also for other mass spec-oriented omics sciences, such as proteomics and glycomics. And I would like to challenge many other fields involved to mass spec, and I believe that the science of computational mass spec is attractive and essential field in biology. And from next slide, I will focus on the topic of plant-specialized metabolite annotations, in which so many complicated natural product structures were the target of computational mass spec. So this complicated slide shows the strategy to characterize metabolomes in various living organisms, and we applied this workflow to a total of 12 plant species in this study. And the first and second steps are constructed to group the ions from one metabolite. It means in the electron spray ionization ESI-oriented metabolomics, so one metabolite can be detected as various ion types, including isotopes, different adduct types and clusters, and ion source fragment ions. And currently, many studies have reported that 90% of detected ions can be recognized as the redundant byproduct ions or noises. So therefore, the important task for unknown metabolite annotation is to determine the peak character and adductor type before the further annotation step is started. And for example, so this slide shows the spectra of kenferol 3 glucoside 7 rhamnoside. So the above two spectra show the MS1 spectrum of positive and negative ion mode at the elution time of this standard compound. And here you can see several big ion source fragment ions like MOVG287 and MOVG433 in positive ion mode. And you can see a big cluster ions in the negative ion mode. And of course, so these cluster ions or basic isotopes and adduct forms can be determined by the basic mass spec data processing technique. But the important things that I want to mention here is that the ion source fragment ions can also be determined by looking at the MSMS spectrum of higher MOVG ion, in this case MOVG 595. So it means, so for example, this is the MSMS spectrum of this metabolite, in positive ion mode, and you can see the simple neutral losses showing hexose and deoxyhexose moieties like this. And the point is, so by looking at this MS spectrum, we can interpret these big ions in the MS1 spectrum as ion source fragment ions of MO-Body 595 because these MO-Body 433 and 287 ions are also monitored in the MS spectrum of MO-Body 595. And furthermore, so we can also determine this adductor type by looking at the mass difference between negative and positive ion mode. And here there is 2 Dalton mass difference between positive and negative ion mode, and therefore the adductor type can be recognized as M plus H and M minus H. And the current MS dial program can automatically annotate these kind of peak characters with the integration method of positive and negative ion mode data to refine the adductor ion hole. And let's move to the third step. And the third step is about MS spectrum searching. And first of all, we analyzed around 500 standard compounds of plant specialized metabolite to annotate metabolite at level 1 annotation, which was defined by Metabolomics Standard Initiative. And we call it as plasma database, and we integrated the publicly and commercially available spectral libraries as much as possible to annotate metabolite as level 2 annotation. And this slide showed the uniqueness of our plasma spectrum databases. In fact, around 166 compounds have unique structures which have never been registered in other databases. And the result of principal component analysis using PubChem fingerprints showed that our plasma database covers a large variety of metabolite structures. And the right panel shows the elution profile and MO-Body value of our standard compounds, and this showed that the plasma database contains so many variety of plant specialized metabolites, which contain the alkaloid and phenylpropanoid flavonoids, steroidal saponin, toliterpene saponin, etc. However, as you know, the coverage of mass spec library is still very small, and actually I counted the unique structures registered in mass spec library and structure databases by using the first layer of InchiQ, and I concluded that the current mass spec libraries covers around 3.7% of structures recorded in the metabolome structure databases. So therefore, we have to fill this large gap by the computational mass spec technique. And the current MS dial program has an isotope tracking function which groups the ions from the same metabolite. And I made this function to deal with many types of experiment, so such as turnover analysis of metabolites in the time course experiment. But we use this function to determine the carbon number of unknown molecules in this study. And actually, so we applied this methodology for a total of 31 plant tissues of 12 plant species. And so once for example, we imported 2 experimental files coming from 12 carbon and 13 carbon plant tissues. So we can look at this kind of blue and red color spot like this. And the carbon element number can be determined by checking the mass shift between 12C data and 13 carbon data. And please make sure that the colors in MS dial were determined by the isotope weight number, and therefore the colors from 12C sample does not always become blue colors. So anyway, the important things that I'd like to say here is, so if you perform the right click in this window, you can call MS Finder program to predict the molecular formula and structure with the information of carbon element number. And the current MS Finder program enabled us to perform the molecular formula prediction, metabolite class, a recommendation, and searching structures of the target molecular formula. And in this study, this program generates the formula candidate with the information of carbon element. And this program assigns the fragment ontologies to each of product ion and neutral losses in the non-biased manner. And I'd like to explain how this fragment ion can be assigned to each MSMS spectrum. And so first of all, so the fragment ion database was created by means of this workflow. So first of all, we extracted a kind of high quality MSMS spectral information having more than less than 10 millidalton mass accuracy and more than 5% relative intensities. Moreover, so multiple spectrum records of the same metabolite coming from different instrument or mass spec conditions are integrated to one metabolite record based on the assigned molecular formula, assigned fragment formula. And the ion intensities are simply assumed because it was not used in this study. And here we could get this kind of structured and MSMS spectrum pair. And we used MS Finder program to assign the substructure form to each product ion and neutral losses. And I just would like to use the first layer of inch key to describe the substructure information from the next step. And now this means, so this structure, so we would like to say this structure may generate these fragment structures. So however, so this kind of dictionary is only available for this compound. So unfortunately, so for example, this dictionary cannot be applied to other flavonol metabolites. So therefore, we tried to convert all of structure information to ontologies. And the fragment ontologies were determined by classifier program from David Wishart laboratory by the input of Smiles code, and metabolite ontology was manually determined by our creation team. And the reason why we manually assigned the metabolite ontology was that, for example, the current classifier assigns flavonol glycoside and flavanone glycoside as flavonoid glycosides. Although we can distinguish these flavonol and flavanone by the MSMS spectrum. So that's why I manually determine the ontology in this study, but so I'd like to say, so MS classifier so is seriously a great tool to improve the metabolite annotation in our studies. And importantly here, our fragment database currently contains around 4,000 queries, in which a total of 459 ontology species are included as fragment ontologies. And moreover, the ontology set so is used to recommend metabolite class for unknown spectrum, and what we call is so fragment set enrichment analysis, FSEA, that I will talk about in next slide. And currently this FSEA set contains around 6,000 query pairs. And again, so one of the reasons why we created this fragment database is to perform metabolite classification. And currently, MS Finder can retrieve structures from various databases, including PubChem and PubMine, Metabolic in Silico Networks Expansion Database. However, especially in natural product chemistry, we will encounter the case that we cannot find any structure candidate from the target molecular formula. And therefore, to increase the information for metabolite annotation, we created one methodology, fragment set enrichment analysis, to recommend the metabolite classes by Fisher's exact test. And the basic idea of FSEA is the same as gene set enrichment analysis. In GSEA, once we uploaded the significant gene list from microarray or RNA-seq results, the significant gene ontology is suggested. For example, a total of 7 genes are involved to the function of fatty acid elongation, and if 4 genes are included in the significant gene list, the p-value is estimated by Fisher's exact test, which considers the total count of uploaded genes. And I translated this idea to metabolite ontology estimation. And for example, if unknown spectrum contains 3 fragments which are involved to the annotation of phosphatidylcholine, the p-value is estimated by Fisher's exact test with the consideration of total spectrum count. And in this study, we defined the significant peaks by the intensity cutoff, and the intensity having more than 5% relative intensities was recognized as significant peaks in the MSMS spectrum. And here, let's consider the total of 10 significant peak features are observed. It contains 5 significant product ions and 5 significant neutral losses. And fragment ontologies are assigned by fragment databases that I present in the last slide to the significant peak list. And the assigned ontologies should include true positive and false positive assignment. And here, as please imagine, the correct ontology of this spectrum is phosphatidylcholine. And therefore, triterpene, cholesterol, flavone ontologies are recognized as false positive assignment. And 3 ontologies, fatty acid, phosphocholine, and choline, which are involved to the phosphatidylcholine annotation, were found in the significant peak list, and 2 of ontologies, so water and rhizopc, were not found. And so it means so 2 by 2 cross table becomes like this. And moreover, 4 ontologies assigned in this MSMS spectrum were not registered in phosphatidylcholine dictionary. and the count is inserted to bottom left panel by 2 by left cross table form. And finally, the count for bottom right panel is so automatically determined by considering the total count of registered ontologies, which currently contains 459 ontologies in our databases. And the p-value is estimated by Fisher's exact test with this 2 by 2 cross table form. And I think, so one of the drawbacks of this methodology is that there is no objective way to define what the significant peaks are. And therefore, I created several methods to define peak significances, but I will perform the further investigation to recommend metabolite classes more effectively. And so this slide shows how users can elucidate unknown MS spectrum. And this is the main window of MS Finder program. And once users double-click this query, this program generates the candidate of molecular formula. And importantly, again, so this program used the information of carbon element count in this study determined in MS dial program, and therefore we can only look at the result of molecular formula containing the specific carbon number, in this case 15 in this example. Moreover, you can find the candidate of structure in the right panel. Here we can see one structure candidate for this molecular formula candidate. And the MS Finder programs also give us the information of metabolite ontologies, which was automatically determined by classifier program. And so moreover, even if we cannot see any structure candidate in right panel, once you click this FSEA button, you can see the result of metabolite class recommendation by FSEA algorithms. And because so this MS spectrum contained the information of 2 fragment ontologies, so sulfate and monosaccharides, which are involved to the glucosinolate class, the FSEA program could recommend so this spectrum as the glucosinolate class. And again, so this p-value is calculated as 2 of 4 ontologies included in glucosinolate class was found in a total of 14 uploaded spectrum. And so as a summary, so our platform can achieve more than 99.8% accuracy for molecular formula prediction when the carbon is fixed. And the program can put the correct metabolite class and structures with 75% and 66% accuracies if the molecular formula has been already registered in structure databases. Also, so these accuracies may depend on the size of database, but we concluded that the accuracy of our platform is very nice to consider the candidate for unknown spectra. On the other hand, the result of FSEA accuracy looks low prediction performance, but I would like to say one compound may be classified to many metabolite classes. It means so we can believe that many metabolites cannot be defined by one metabolite class. And therefore, I would like to emphasize that so FSEA is not for the accurate metabolites class prediction, but for the recommendation tool to know the metabolite class candidates for unknown MS spectrum. And importantly, all these slides show the accuracy of structure elucidation is not perfect. So it's not reach to 100% accuracy. And therefore, so we have to fill this gap to achieve more accurate prediction of structures. And therefore, we also integrated the idea of molecular spectrum networking as the GNPS does in our study. And all of metabolite ions are connected by 3 ways, including MS-MS similarity, metabolite ontology similarity, and bioreactivity. And this slide explains how our plant metabolome network was used for metabolite annotations. So each node describes one detected metabolite ion. And the node color describes the plant origin of detected peaks. For example, these metabolites were colored by green because they were only detected in the Arabidopsis, and the nodes are connected if the MS-MS spectrum was similar between 2 metabolite ions, and the node were also connected if the metabolite ontology names are similar. And for example, so this metabolite was originally unknown, and the metabolite was connected to the glucosinolate metabolite by the MS-MS spectrum similarity, and therefore we could conclude that this metabolite may contain the moiety of sulfate and hexoses. And we could finally determine this metabolite as hexosylvanillic acid sulfate. And by the same manner, this metabolite was classified to 12 sulfooxyjasmonic acid. And then this kind of network function was also implemented in MS-Dial and MS-Finder programs. Moreover, we organized the characteristic ion and the neutral loss features, which should be monitored in MS-MS spectrum to characterize unknown spectrum to the specific metabolite class. And this table contains the name of metabolite class, the major plant species, and the detectable adduct types, and unique ions and neutral losses in positive and negative ion mode data. And we organized this kind of characteristic ion table for around 80 specialized metabolite class. And finally, we could create this kind of figure, and x-axis is the retention time. The upper area of y-axis shows the MOVG value of annotated metabolite in positive ion mode, and the lower area of y-axis shows the MOVG value of characterized metabolite in negative ion mode. And the spot color describe the metabolite classes. And blue colors of structures mean the characteristic product ion, which should be confirmed to characterize metabolite class species. And red color of structures mean the neutral loss, which should also be confirmed in the MS-MS spectrum for metabolite class characterizations. And finally, we could annotate more than 1,000 metabolites in a total of 12 plant species. And in addition to well-characterized flavonoids, glucosinolate, and phenylpropanoids, our methodology covers various metabolite classes such as striatal saponin and triterpene saponins and beta-carbolin alkaloids and something like this. And finally, so we applied our methodology to clarify the metabolome of 25 natural variants of Arabidopsis, and these natural variants can be classified by the flowering time. And by using around 300 annotated metabolite information, we concluded that the flowering time can be explained by the metabolome of shoot and root parts of Arabidopsis. And furthermore, we investigated the importance of plant specialized metabolite for several biological phenotypes. And this circus plot showed the summary of our metabolome data, and circus was separated by the metabolite classes. For example, the first part contains amino acid, and the second part contains information of flavonoids. And the second layer showed the heat map of 25 natural variants, and the third layer indicated the size of ion abundance. And furthermore, the bar chart means the importance of metabolite associated to the flowering time phenotype. And fifth layer shows importance of metabolite associated to the various physiological traits and phenotypes for nitrogen depletion and drought stress. And so, for example, in shoot, the glucosinolate metabolite substantially changed among 25 natural variants, and these are highly correlated to each other. And moreover, the glucosinolate metabolite can be considered as the relevant metabolite with various traits and phenotypes. And in root, the relatively rare sulfate conjugated lignols, guaiacylferric acid sulfates, were substantially changed among accessions, and we could also find many metabolites relevance with the trait, such as dicoumaloyl spermidine and total plant height. Importantly, this slide showed that our methodology can be used not only for metabolite annotation, but also for the detailed investigations for plant physiologies with information-rich metabolomics data. And finally, all of our database and programs are shared in our Recam Prime website, and you can search your unknown query in also our newly created web server. And so all of the details that I introduced today was summarized in our latest paper. And again, thank you so much again for all of collaborators to contribute to this research. And okay, I finished my talk today. And lastly, I'd like to say I'm sorry for all of users for giving us the feedbacks. And I know that I don't take care of many feedbacks. Actually, I cannot take care of many feedbacks, but I actually remember all of your feedbacks, and I would like to deal with your feedbacks as much as possible in future. And actually, I appreciate all of your always feedback to improve our program, and I always welcome for your effective suggestions or comments. And hopefully, I'm looking forward to meeting with you at the heart of this year. And thank you very much for listening my talk. And I'm very welcome your questions or comments. Thanks.
Well, thank you very much, Hiroshi, for this great talk, and thank you for giving it so early for you. As you know, it was not easy to find a time that would be convenient for everyone. If you have any questions, you can type them either in the chat or in the question box, and Dr. Hiroshi Tsugawa will answer them live. So we are just gonna wait a few minutes to see if you have questions for him.
Hi, Elena. I think I will start with a couple questions that are already in the chat box. And the question is from Santosh Kumar. And he asks, do you need both positive and negative ion mode for the MS dial to process the data? And also, do you need MS of both the positive and negative ion mode? That's his first question.
Can I answer this now? Oh, all right. So
yes, go ahead if you can.
So actually, MS dial doesn't require the MS spectrum information for this function. so because MS dial is actually constructed for for any type of mass spec data, but I just said so the accuracy will be increased if the MS spectrum is available.
Okay. The next question is, can we use iron 59 isotope feeding type experiments with
So internally, we have 15 nitrogen and 34 sulfur fed plants data. But so you said 14 carbon or 15 nitrogen? So you said. But so anyway.
It's iron 59, yeah. But
so if you see the MS dial option button, so now I'm preparing several options capable of 13 carbon, deuterium, 15 nitrogen, and 34 sulfate, and such combinations.
okay.
Okay.
So maybe in the future you'll be able to do some iron isotope feeding type experiments, but not quite at the moment. Okay, next question comes from Biswapriya Mishra, and he asks, does MS dial penalize mismatches, and if so, how?
So again, excuse me?
I'll reread. Does MS dial penalize? Is there like a negative impact if there's a mismatch? And if it does so, how does it do that?
So I'm not sure if I can correctly answer this question. So I know the combination of MS dial and MS finder program does not always annotate the metabolite correctly. And therefore, at least in this study, so that's why I integrated the several methodologies, which also contain the molecular spectrum networking. And so also we made a kind of the characteristic ion database to confirm the metabolite classes. And so that's why we, in this study, confirm every peaks manually. So to accurately say, we believe this MSMS peak is this guy, something like this. And so for example, if we can see the ions in both positive and negative ion mode, it was easier. But sometimes we could find the peak in either positive and negative ion mode. So that it was a difficult case, and we are actually very struggling to annotate such kind of metabolite.
Okay, and switching gears a little bit, this question is from Arvind Subaraj, and he asks, other than lipid maps, are there any other databases for MS2 lipid annotation?
For lipids?
Yes.
So now actually MS Finder incorporates the lipid maps only for lipids. But so of course in future I will integrate several lipids database, for example lipid home, and for example lipid match or several lipid consortium database, because the recently, so from last year, new initiative, Lipidomics standard initiative was also started. And so my professors Makoto Arita and Masanori Arita are belonging to this initiative. So that's why I will communicate them, and I will integrate several databases if needed.
Okay, and another question, again for MS Dial, from Yassine Al-Abedi. He asks, how is MS Dial dealing with chimeric spectra, that is 2 or more precursor ions in one MS2 scan?
And again, so you mean how MS Dial deal with what?
Chimeric spectra, that is 2 or more precursor ions in one scan.
Okay, so I'm not sure if I can answer this question correctly. So currently, MS Dial just suggests so this guy and this guy should be coming from the same metabolite, and MS Dial doesn't completely the group of such ions. And I personally use the bigger ion abundance one. So if there are 2 peaks existed, but so it depends on the purpose of metabolomics. For example, when I'd like to do the annotation of new metabolite, so the MSMS spectrum of natrium or sodium and calcium adduct, something like this. So the few MSMS spectrum information is observed usually compared to the spectrum from M plus H, M minus H, or so formic acid adduct or acetic acid adduct. So that's why I will use the M plus H and M minus H spectrum for the annotation purpose. But so I would like to do the quantification of metabolite, I sometimes use sodium adduct type, something like this.
Okay, there's one more question, and it's a general question, is can one use MS Finder standalone for formula searching with only MS information?
Sorry, I couldn't receive the final voice because there are some noises.
Okay, so the question is, can one use MS Finder as a standalone platform for formula searching with only MS information?
Sure. If actually there is a tutorial in the website, and actually all of my programs is available as a standalone software. But so I just want to say, so there are also the linkage among the software that I've developed. And so MS Finder definitely can import only MS1 spectrum for molecular formula prediction.
Okay,
all right, so I think
answered the questions.
Yeah, I think we're good. I'm just gonna introduce you the next webinar if we're done with questions. So stay tuned for our next webinar, the 24th May, which will be hosted by Fatima Bindewala, and we will be featuring Dr. Robert Spores, and he will talk about combined NMR and MS Metabolomics to study neurodegenerative disease. Again, thank you very much for attending this webinar, and thank you, Dr. Hiroshi Tsugawa, once again, and have a good day or night for wherever you are.
All right, thank you so much from me as well.
Thank you.